Hello, everyone, and welcome to today's webinar titled Detection of SARS-CoV-2 Neutralizing Antibodies with GenScript's SARS-CoV-2 Surrogate Virus Neutralization Test. This webinar is part of the 8th Coronavirus Virtual Event Series, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to genscript.com. Now let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located on the left of your screen. You can also participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. Now, if you have any trouble hearing or seeing this presentation, simply click the desk button located at the top of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on that continuing education credits link located in the abstract window below and follow the process to obtain those credits. I now present today's speaker, Amanda Grimm, a senior product manager at diagnostics at GenScript USA. For a complete biography of all of our speakers, please visit the presenter tab from the menu at the left of your screen. Amanda, welcome. You may now begin your presentation.
Hello and welcome. My name is Amanda Grimm, and I'm the Senior Product Manager for Diagnostics here at GenScript. In today's presentation, I'm going to talk about the detection of SARS-CoV-2 neutralizing antibodies with the surrogate virus neutralization test. We have three main takeaways from this talk. The first is going to be principles of the CPAS surrogate virus neutralization test, otherwise known as the CPAS sVNT, and how it compares to the gold standard live cell virus neutralization test. The second being neutralizing antibodies as correlate of protection for adaptive immune response, and the last one using the sVNT to determine neutralizing activity against the variants of concern. So this is just a quick overview. We can take a look at where we are number-wise since the pandemic started. As of early October, the WHO reports over 615 million infections, 6.5 million deaths, and the numbers do continue to rise. Let's review how SARS-CoV-2 infects the cell. Here we have a representation of the virus and its structure. Over here on the outside of the virus, it's peppered with the protruding spike protein, and then on the ends of the spike protein, we have a region called the receptor binding domain, or the RBD. In order for infection to take place, the RBD binds the ACE2 receptor on the lung epithelial cell. This protein-protein interaction is key to virus propagation, so by blocking this, also called neutralization, the virus cannot invade. It does not propagate, and disease is prevented or reduced. How does the CPAS kit work? The CPAS technology is based on detecting neutralizing antibodies that block virus infection, as we just went over in virus infection of the cell. The kit is a competitive ELISA platform whereby the ACE2 protein is coated on the bottom of the plate wells. Samples are incubated with RBD HRP so that any neutralizing antibody present will bind the RBD HRP. When this neutralization solution is added to the plate, this complex inhibits the ability of the RBD HRP to bind with ACE2, so that samples that do not contain neutralizing antibodies, those wells will result in color development, whereas those containing neutralizing antibody will inhibit, resulting in little to no color development. How does this compare to conventional VNTs? VNTs require strict safety conditions to grow the biologicals associated with the assay. This process is time-consuming, taking days to grow cells and virus, and then it also requires someone with high expertise to read the test. Our test can be run in most laboratory settings, making it faster and higher throughput. This is capable because now you have 96-well plate, and this can also be automated. The commercialization of this also means that testing is more consistent from sample to sample. This study is from early in the pandemic and demonstrated neutralizing antibodies as a correlation to immunity. The study also used our sVNT, and they looked at an outbreak on a fishing vessel. What they found was a small group that were seropositive before departure, and these individuals did not experience symptoms or disease while the outbreak spread. It was concluded that the presence of the neutralizing antibodies from their prior infection was associated with protection against reinfection. It's important to note that not all antibody tests on the market detect or can differentiate neutralizing antibodies from binding antibodies. The GenScript test is unique because it confirms neutralizing function and because of its technology is also isotype and species independent. Other tests capture binding antibodies that may be binding any epitope of the RBD or antigen used on the plate, but it does not provide results for the ability to neutralize the virus. Early studies demonstrated poor correlation of regular antibody assays with neutralization activity. This publication here compared 3 commercial antibody assays where 2 of the serology assays were against the nucleocapsid protein and the third against the spike 1 protein. When compared to focus reduction neutralization tests, this is specific for neutralizing antibodies, we see a very low correlation result represented by the low R-squared values. We also see that positive binding in ELISA may not mean positive neutralization, as discussed in this publication. The data generated from this study was collected from a total of 382 Navy service members aboard an aircraft carrier during a COVID outbreak. During this event, 228 participants had positive ELISA results, and of those, a little over half also had neutralizing antibodies when tested in the microneutralization test. GenScript's sVNT assay achieved multiple regulatory approvals, including the US FDA Emergency Use Authorization, the CE mark in Europe, and the Health Science Authority approval in Singapore. Our kit comes with all the necessary reagents to perform the assay. The format of the kits are one-plate kits. In November 2021, we received an updated authorization for the kit for semi-quantitative use, in addition to the qualitative option. This data was submitted as part of our FDA review, and this particular chart is a summary of our original sensitivity specificity data when compared to plaque reduction neutralization test. You can see that our test performs very well, resulting in 100% for both sensitivity and specificity. In this slide, we also show where we conducted a testing timeline to compare to PCR. As expected, early in infection, concordance is little to none, but as time goes on and the immune responds, the sensitivity increases, which looks optimal by day 8. The semi-quantitative results can be reported with a numerical units per mil for positive samples that are over 30% inhibition. The range includes an upper and lower limit of 185 and then 47 respectively. This interpretation is included in the IFU. We were also able to conduct and include additional analysis showing concordance between CPAS and the titers obtained with PRNT50. You can see here in the red where we have identified high and low titer being above and below 100 units per mil. I just talked a little bit about the sVNT and its relation to titer levels. So what does that mean and why is that important? Identifying titer levels has been used to establish vaccine efficacy during COVID vaccine development. This study in particular with Peter Gilbert and David Montefiore is a coronavirus efficacy phase 3 clinical trial for the mRNA 1273 related to vaccine recipients that were assessed for neutralizing antibodies as well as binding antibodies, as these are potential correlates of risk and protection. Discussed in the paper, they found vaccine efficacy is upwards of 98% at the high titer level. They also found that neutralizing antibodies mediate approximately 2/3 of vaccine efficacy, so pretty important. The estimated risk was also about 10 times higher in those recipients with low or undetectable levels when compared to those with antibodies in the top 10%. Now, building off neutralizing antibody titers for vaccine efficacy and correlates for risk and protection, another study here by Miles Davenport's group additionally adds support to neutralizing antibodies as a biomarker, and that this could be used to determine when boosters could be taken. In the study, they generated a model based on neutralizing antibody levels in order to predict what this timeline would look like. The top figure here shows that when protective efficacy starts at 90%, the previous study, if you remember, indicated this to be considered high titer level. The model here predicts that by 201 days, that's 28 weeks or about 7 months, 70% efficacy will be reached, which is still really good. 50% isn't predicted to be reached until after 250 days, which is about 35 weeks or almost 9 months. SARS-CoV-2 is a rapidly mutating virus, and we've seen a number of variants emerge in the last couple years, with the most recent variants being from the Omicron group. Here at GenScript, we continue to support the fight against COVID-19 and have quickly made variant-specific RBD HRP reagents available for those interested in variant-specific studies. The RBD HRP that comes standard in the kit is wild-type based, and this can easily be swapped out for one of the variant reagents specific to the investigation. These reagents are provided so that equivalent dilutions are made as you would use the component that comes standard in the kit. This was a study in Thailand looking at solid cancer patients and their immune response to the AstraZeneca vaccine when compared to healthy individuals. Their study goals were to generate more data to better determine next booster timeline and to assess the vaccine efficacy in cancer patients. They found the total immunoglobulin decay rates to be similar to healthy individuals, even though they averaged a lower overall response. In addition, they used the GenScript sVNT and Omicron RBD HRP to evaluate the ability of these patients to neutralize the Omicron variant. Their findings for this case was that the neutralization ability was deemed poor even in those considered to generate a high titer. In this slide, I'm sharing one of our US customers that conducted an opt-in participant study looking at variant neutralization across vaccination, infection, and hybrid of infection and vaccination. The data is compiled into a public dash, shown here, where you can see the resulting hybrid combo provides better neutralization capability against Omicron. And this would have been early Omicron, so BA1. If you want to look at this more in depth, you can visit the CureHub website listed on the slide to look more at the data they collected. So the summary of information that we covered in this presentation is that CPAS sVNT is the best choice for alternatives to the long and time-consuming live virus assays for detecting neutralizing antibody. The presence of binding antibodies may not guarantee presence of neutralizing antibodies, and those levels may not be consistent. Neutralizing antibodies are an important biomarker for adaptive immunity, and that variant of concern studies offer variant neutralization insight, especially into those considered high risk. And with that, I want to say thank you for your time and attending this presentation. If you have questions, you can email us at diagnostics@genscript.com.
I want to thank you, Amanda, for your time today and for your important research. And before we go, I want to thank our audience for joining us today. And I'd also like to thank our sponsors, GenScript, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until October 19th, 2024. Laborers will alert you via email when it's available for replay. Now, we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and bye-bye.